uh, 3,000 to 3,000, 30,000 by size. These mechanical methods bring the non-specific breakages in DNA and the resulting fragments are mostly blunt-ended. Shotgun method. These are also one type of the isolation, the selection of DNA. The shotgun method is it involves the restriction digestion of cellular DNA by using the specific restriction in endonucleases, generating variable shotgun method. It involves the designation of the cellular DNA by the using specific restriction endonuclease, generating the variable DNA fragments. These DNA fragments are cloned into the suitable vector to produce the RNA, which are the transformed into the recipient, the bacterial clones. This each and the every DNA fragment, cellular DNA, is cloned in the from the bacterial clones. The bacterial clones are the screen to isolate and desired gene. This technique is also known as the shotgun cloning. Shotgun method. RDNA directed synthesis. Desired DNA can be synthesized from the mRNA by the process of called reverse transcription. This the discovery of the enzyme reverse transcripted by the terminant the Baltimore from virus made to the possible alter the central dogma in molecular biology. The the DNA synthesized from the mRNA is known as the complementary DNA or cDNA. These are the isolation method. Next one is the selection of the suitable cloning vector. The first one is isolation, and next one is the selection. Mainly, a cloning vector is a self-replicating DNA molecule, into which the desired DNA is to be integrated. A suitable cloning vector is the selected in the next step of rDNA technology. Most commonly uses the vectors or plasmids and bacteriophages. The next technique is also introduction to desired DNA into the vector to form RNA molecule. The desired DNA, the desired DNA or the DNA insert which has been extracted and cleaved enzymatically by the selective restriction endonuclease enzymes are now ligand by the enzyme ligase to the vector DNA to form a RNA molecules, which is often called the cloning vector desired DNA construct. RNA molecule is introduced into the suitable host. Suitable host cells are the selected and the RNA molecules are formed introduced into these host cells. This process in the entry into the RNA into the host cells is also called as the transformation. Usually selected hosts are the bacterial cells like E. coli, yeast, fungi may also be utilized. Next one is the next process, the selection of transformed host cells. Transformed cells are those cells which have take up the RNA molecule to be separate from the non-transformed cells using the various methods, making is the marker genes. Next one is the expression and multiplication of the desired DNA in the host cells. Now, finally, it is to be the ensured that the foreign DNA inserted into the vector DNA expressing the desired character in the host cells. Also, the transformed host cells is the multiplied obtained the sufficient number of copies. If needed, such genes may also transform and express into the another organism. These are the techniques in isolation. What are the enzymes are used in the DNA technology? Enzymes used in the RNA technology. What type of the enzymes are used in the restriction enzymes, exonuclease, ligase, polymerase enzymes? These are the enzymes are used in the RNA technology. What are the restriction nuclease? Gene cloning requires the DNA molecules to be cut the very precious and the reproducible fashion. This is a precious cut in the both donor and the recipient. DNA is achieved through a set of enzyme is also called as restriction endonuclease. These enzymes occur naturally in majority of the bacterial species to restrict the replication phases through the DNA degradation. Restriction endonuclease, popularly called also molecular seizure, of the immense is the molecular biologist cut the DNA in the precise manner. These enzymes serve the important tools to cut the DNA molecules in the specific sites, which is the basic need for the DNA technology. These enzymes are that produce the internal cuts in the strands of the DNA only within or near some specific sites is also called as the recognition sites or recognition sequences or restriction sites or target sites. Such recognition sequences are the specific for the each restriction enzyme. The presence of the restriction enzymes was the first of all reported by the W. Arbor in the 1962. He found that when the DNA is phase introduced in the host bacteria, it was fragmented into small species. This led him to the postulate the presence of the restriction enzymes. The first true restriction endonuclease was the isolated in 1970s from the bacterium E. coli by the Melson and Yoon. Another important breakthrough was the discovered the restriction enzyme is the HIN2 in 1970s by the Kelly and Smith and Nathans. The isolated from the bacterium Haemophilus influenza in the year 1978. He got the Nobel Prize in Physics Medicine by the Smith, Arbor, and Nathans for the discovery of the endonuclease. What are the types of the resistant endonuclease? These are the three types of the resistant endonuclease: type 1, type 2, type 3. Type 1 is the mainly they are the complex type of the endonuclease which cleave only one strand of DNA. These enzymes have the recognition sequences of about 15 base pair length. They require magnesium ions and ATP for their functioning. The types of the restriction endonuclease cleavage DNA about the 1000 pairs away from the 52 end sequence TCA located within the recognition set. This is cycle located within the recognized set. Restriction two type of the type 2 restriction endonuclease. These are the most important in endonuclease for the gene cloning and hence for the RNA technology. These enzymes are the most stable. They show the cleavage only at specific sites and therefore they produce the DNA fragments in defined length. These enzymes show the cleavage in both strands of DNA immediately out within the recognition sequences. They require magnesium ions for their functioning. Such enzymes are the advantage because they don't require ATP for the cleavage and they cause cleavage in both strands of DNA. Only type 2 restriction nucleases are used in the gene cloning or DNA technology due to their the suitability. The recognition sequence for type 2 restriction nuclear enzymes are the form of the palindromic sequences with the rotationally symmetry. The base sequence is the first half of one strand of DNA is the mirror image of the second half of their other strand of the DNA double helix is also called as the palindromic sequences. Important examples of the type 2 restriction endonuclease include the in Fi E. coli, Ri, Pu2, allular, etc. Some of the enzymes. Next one, type 3 restriction endonucleases these are the most these are the not used for the gene cloning techniques. Third type. The second one is the very important type of the restriction endonuclease. The third one is that there is no important in gene cloning technique. They are the intermediate enzymes between type 1 to type 2 restriction endonuclease. This require the magnesium ions and ATP for the cleavage and the cleave the DNA well defined sites in the immediate vicinity of recognition sequences in of the nature of the cleavage produced by the Christian endonuclease is the considerable importance. They cut in the DNA molecule in the two ways. Mainly, many restriction endonuclease the cut off the DNA molecule into two ways. One is the many restriction endonuclease cleaves both strands of DNA simply the same point within the recognition sequence. As a result of this type of the cleavage, the DNA fragments with blunt ends are the generated. Huviol is the example of the restriction endonuclease producing the blunt ends. Blunt ends may also be referred to the flush ends. The action of the small. The second one is the another way is the second way is the cut of the DNA. In this style cleavage of the restriction endonuclease, the two strands of DNA are the cut to two different points. Such a cut points are the length, the staggered cuts, and this is the result in the generation of the protruding ends. One strand of the double helix extend a few bases beyond the other strands. Such ends is called as the coincidental sticky ends. Nomenclature. Nomenclature also the discovery of the large number of cysteine nucleases is named by the standard procedure. A suitable system is proposed by the Smith and Nathans in 1973. The first letter in the italics in the enzyme indicates the genus name. For example, hin is the followed by the for example first two letter also in italics the species, then comes the strain of the organism, and finally Roman nomenclature or a couple of the examples given below the E. coli. E. coli is a species, or is the from the E. coli strain RY13 or Echerichia E. coli co-species. Genus is the Echerichia, coli is the species. The strain R is the RY13. The first endonuclease one is to be discovered. In 3 is the from the Haemophilus influenzae, the strain, and the third endonuclease to be the discovered. The next enzyme is the exonuclease. Exonuclease is the enzyme that removes the nucleotide from the ends of the nucleic acid molecule. The exonuclease removes the nucleotide from the 52 or 32 end of the DNA molecule. An exonuclease never produces the internal cuts in DNA. In RDNA technology, various type of the exonucleases are the employed, like the exonuclease ball 31, E. coli exonuclease 3, lambda exonuclease, etc. Exonuclease ball 31 are the employed for the making the DNA fragment with blunt ends, the shorter from the both ends. E. coli exonuclease is utilized for the 3-end modification because it has the capability to remove nucleotides from 32 OH and the DNA. Lambda exonuclease, it is the modified to 52 plus ends of the DNA is the removes the nucleotides from 52 turns in inner linear DNA molecule. DNA ligase, the mode of the action of ligase is the opposite to nuclease. Ligase and 5 prime and 3 prime is the OH is the two nucleases to establish phosphodiester bond between them. Thus, they capably joining together two individual fragments, the double-stranded DNA. The process is also called as the ligation. All living cells produce the ligases, but the enzymes use the genetic engineering are usually purified giant two fragments of DNA by synthesizing the phosphodiester bonds. They function to repair the single strand nicks in DNA double helix and in rDNA technology are the employed for the sealing the nicks between the adjacent nucleotides. This enzyme is the terminated in the molecular glue, DNA ligase. The polymerase, the DNA polymerase is the enzyme that is the synthesize new strands of the DNA complementary to an existing DNA or RNA template. DNA polymerase can function only if the template possesses a double strand region that act as the primer for the initiation of the polymerization. Three types of the DNA polymerase is the routinely in the genetic engineering. One is the DNA polymerase one. This enzyme attaches the short single strand region or neck in a mainly double strand DNA molecule and then synthesize the complementary new strand, degrading the existing strand as it is the proceeds. Thus exhibits the dual activity DNA polymerization and DNA degradation. The Kellner fragment, it is the DNA polymerase 1 without the exonuclease activity. It synthesizes the complementary DNA strand and a single strand template. The major application of the Kellner fragment and related to the polymerase are in DNA sequencing. Reverse transcriptase. In nature, this enzyme is involved in the replication of some RNA viruses. These are nature enzymes involved in the replication of some RNA viruses. The enzyme synthesis DNA strand using the RNA is the template. This ability to synthesize DNA strand complementary to RNA. Template the central to the technique is also called as the complementary DNA cloning. These are the RDNA technology techniques, isolation. Next one is the some of the enzymes are involved in the RDNA technology. Exonuclease, DNA ligase, trypsin endonuclease. Next topic is the methods of gene transfer techniques. Mm, methods of the gene transfer. And genetic transformation is the powerful tool and the important technique for the production of the transgenic plants. This is useful to introduction of the foreign genes into the crop plants, mainly the creating of the genetically modified organisms. Mainly these are the plant breeders and the genetic engineers share the common goal of plant improvement, while the plant breeders traditionally use the selective breeding for Peritial enhancement of the genetic engineers continue to develop the techniques for the isolation and the insertion of gene for desirable traits. In this technique, genes which are the unavailable to the particular plant species may be obtained from the other organism using the both molecular and the cellular biology techniques. The transfer of desirable genes from one organism to other organism is and expression is the foreign gene in the genome referred to the genetic transformation. The transfer of gene is known as the transgene or the and the organism developed from the successful gene transfer is known as transgenics. The plants developed from the suitable integration foreign integration of the foreign gene is known as the transgenic plants. Plant transformation is the process by which is DNA is introduced into the Plant cells are the tissues from the virtually any source. Gene transfer methodology has become the part of an essential technology to manipulate plants for the both scientific and commercial purposes. Transgenic plants, the produce of this methodology is used for the dissecting the mechanisms of plant and gene regulation. This technology is also useful for the identifying and the evaluating the agriculturally useful traits and transfer them into the commercially valuable crops. Plant transformation was first described in the tobacco plants in 1984. The rapid developments in the transformation technology have resulted in the gene modification of many plant species. Various methods for the introducing the diverse genes into the plant have been developed. All the transformation techniques involve the uptake and then in the delivered into the plants. Gene transfer methods in plants make use the variety of target cells types, which is the including the protoplast, or cultured cells, meristem cells, mature embryo, and pollen grain. What are the types of the gene transfer methods in plants? These are the two types of the gene transfer methods categories. One is the direct or vector-less gene transfer method, or second one is the vector mediated gene transfer method. Vector or vectorless DNA transfer methods. The term direct or vectorless transfer of DNA used in the foreign DNA directly introduced into the plant genome. Direct DNA transfer methods rely on the delivering active DNA into the plant cells. This is in contrast to the agrobacterium or vector-mediated DNA transfer, which may be the regarded as indirect methods. Majority of the direct DNA transfer methods are simple and effective. In fact, the several transgenic plants have been developed by this approach. These two types of the physical or chemical gene transfer methods. The physical gene transfer methods are the electroporation, particle bombardment method, next one microinjection, next one is the lipase mediated transformation, chloroplast mediated transpiration, and silicon carbide fiber meter transformation. These are the all are the physical methods. Mainly physical method electrophoration introduce the foreign DNA into the living cells by the exposure of brief electronic pulse is known as the electrophoration. In this techniques, mixture containing cells and DNA is exposed to very high voltage electrical pulses, approximately 4,000 to 8,000 volcanic centimeters for every brief time periods, a few milliseconds. It results the formation of the transient force in the plasma membrane through which is the DNA seems to the enter inside the cells and the nucleus. These techniques can be used for the delivery of DNA into intact plant cells and the protoplasm. Particle bombardment gun ballistics, the microprojectile. It is also called as the particle gun or shotgun or microprojectile gun and the ballistic missile. Ballistics is the combination of the biological and the ballistics. In this method, foreign DNA containing desired genes are coated onto the surface of tungsten or gold particles and are carried by the Marco carriers. These Marco carriers are inserted in the apparatus and bombarded in the high volcanicity onto the plant cells or tissues. With this, the DNA segments are released, which enter the plant cells and integrate with the genome. Next one is the micro injection. Micro injection involves the direct mechanical introduction of DNA into the nucleus or cytoplasm using the glass microcapillary injection. Micro injection. Uh, this technique target cell is poisonated under the microscope and DNA is then directly injected in the cytoplasm or the nucleus with help of the two micro manipulators. One micro manipulator holds the pipette and the another holds of the micropapillary needle. While the gene transfer is done, the recipient cells are kept immobilized in agarose embedding and held by the succussion holding pipette. Once the genes is delivered in the injector cells, are then the cultured into the in vitro regenerated into the transgenic plants. The stable transformants can be achieved through the method, but the require technical expertise and time the consuming process. Also, micro injection has achieved only limited success in a plant transformation due to the check the cells, the plants and the viability lack of availability single cell to the plant regeneration system, the most plant species. Liposome mediated transformation is another way. Liposome is artificially created the microscope. Next is the chemical gene transfer methods. Then we can use the polyethylene glycol (PEG) mediated transfer. Polyethylene glycol is the mediated transformation is method used to the delivery DNA using the protoplast as explants. PEG is the presence of the divalent cations distillables the plasma membrane of the protoplast and renders of the removable and naked genome. The method is the similar to electroporation in that the DNA to be introduced. It is simply mixed with the protoplast and the uptake the DNA stimulated the addition of the PEG is the rather than the electropulse. PEG mediated transformation is the several advantage because it's the easy to electrical pulse. PEG mediated transformation has the several advantages because it is the easy to handle and so specialized equipment is required. Mainly is the this technique is really used to the low frequency transformation. Many species can the degrade. Next one is the calcium phosphate precipitation mediated transfer. The DNA is allowed the mix with the calcium chloride solution and the isotonic phosphate buffer to form DNA calcium phosphate precipitate. When the activities the dividing cells in culture are exposed to this the participate the several hours, the cells get transformed. The success of this method is the depend on the high concentration of DNA and the production of the complex precipitate addition of the dimethyl sulfide DMSO increase the efficiency transformation. Vector mediated are the indirect gene transfer methods. The above is the directed gene transfer methods. Chemical are the physical. Chemical method mainly they can use the PEG. The physical method is the electroporation, shotgun method, different types of the methods. Vector mediated or indirect gene transfer method is the agrobacterium mediated gene transfer in plants. The agrobacterium is known as the natural genetic engineering of plants because these bacteria have the natural ability to transfer tDNA of their plasmids into the genome. These bacteria enter into the plants, into the wound site in the cause of the unorganized growth of the cells is mass known as the crown gall. TI plasmids are used the gene vectors for delivering target plant cells and the tissues. The foreign gene is the cloned in the tDNA region of TA plasmid in place of the unwanted sequences. Agrobacterium tumefaciens is a soil-borne and the gram-negative bacteria. It is a rod-shaped and motile, belongs to the bacterial family Rhizobiaceae. Agrobacterium tumefaciens is the phytopathogen and is known as the nature the most effective plant genetic engineer. In fact, the major credit for the development of plant transformation techniques goes to the natural unique capability of a tumefaciens. The agrobacterium system was the historically the first successful plant transformation system, making the breakthrough in plant genetic engineering. The success in the gene transfer has been achieved by the characterization and exploiting the plasmid carried the bacterial pathogens. These two types of the plant pathogenic bacteria, which use the natural gene transfer agents in plants, agrobacterium tumefaciens that induce the crown gall diseases. The agrobacterium residences, it induces the air root diseases. Crown gall disease, agrobacterium tumefaciens, TI plasmid. Almost 100 years ago, Smith and Townsend postulated the bacteria was the causative agent for the crown gall tumors, although the importance was recognized much later. Agrobacterium tumefaciens infects wounded and damaged plant tissues, induce the formation of tumor called ground gall tumor. In the entry of the bacterium, the plant tissues is facilitated by release certain phenolic compounds by the wounded sites. Crown gall formation occurs in the bacterium visits the TI plasmid to the plant cell cytoplasm. A fragment of TI plasmid, referred to as tDNA, is actually transformed from the bacterium into the host, where it gets integrated into the plant cell chromosome. Crown gall disease is a naturally evolved genetic engineering process. Organization of the TI plasmid agrobacterium tumefaciens. The TI plasmid exists the independent replicating circular DNA molecules within the agrobacterium cells. TA plasmid agrobacterium is the circular DNA molecules up to the 200 kilo base pair in length. The tDNA is the variable in length in the range of the 12 to 24 kilo base pairs, which depend on the bacterial strains. From TA plasmids come Napoleon strains of TA plasmid have one tDNA with the length of the 20 kilo base pair, while octopine strains have to the 2DNA 41, 41 regions referred to the TA1 or TA or the respectively 14 kilobase and the 17 kilobase length. TA plasmid is the major regions for the virulence, origin of the replication, oncogenicity, and the catabolism of opinions studies on A. The tubifensis mediated gene transfer system have the relieved the required DNA transfer of in the plants. The genes present in the tDNA region, the tDNA genes are the biosins of the oxygens, cytokinin, opinions. Okay, virulence region, opinion, catabolism region, tDNA transfer and the integration of the in the host cells. Mainly, these are the tDNA transfer integration to the plant host genome is the describes mainly the below the techniques are also described. Signal induction of the agrobacterium, attachment of the agrobacterium to plant cells, production of the valency proteins, production of the tDNA strands, transfer of DNA in agrobacterium, transfer of tDNA into the plant cell integration. Hairy root disease, the agrobacter rhizogenesis in the plasmids. These are the mainly agrobacter rhizogenesis is known as the rhizobium rhizogenesis. Can also infect the plants and cause the hairy root disease in plants. The plasmids of the agrobacter rhizogenesis have been isolated and the characterized. The plasmid of the agrobacter rhizogenesis is known as the RI plasmid or root-inducing plasmid. Some of the RI plasmid strains possess genes that are the homologous to TA plasmid. Example, auxins, biosynthetic genes. Agrobacter rhizogenesis is a gram-negative soil bacteria. Infects plants. Adventitious roots is also called as the hairy roots. Are induced from the infected site. This event occurs due to the transfer of the particular DNA region called transfer, compressing the loci between the TRTL regions of the root-inducing array plasmid of the bacterium to plants genome. Instead of the virulence gene, RA plasmids contain the series of the opinion regarding the frames of the tDNA or tDNA of the agropine RA plasmid consists the two copies of left TL DNA and the right TL tRDNA. Each copy is transferred independently. A total 18 open regarding the frames was identified the left copy of DNA. It was shown that for the development of the hairy root disease, the presence of the four role genes critical. And next, the genes are the correspondent to regarding the frames. The products of these genes are the involved in the metabolism of plant growth regulators, which gets sensitized into auxin to lead the root formation. Vectors of the rhizogenes is a agrobacterium prevents vectors can be constructed by using the agrobacter rhizogenesis. The agrobacter rhizogenesis mediated transformation of plants can be used the similar manner to that used the during the agrobacter events transformation. Hairy root syndrome and the production of the transgenic hairy roots cultures by the genetic manipulation can be achieved agrobacter rhizogenesis mediated transformation for the transformation. The border sequence of the tDNA are required and the remaining T for the transformation, the border sequence tDNA are required and the remaining tDNA can be replaced with the foreign DNA. After the replacement, whole plasmid the introduced into the cells from which is the whole plants can be regenerated. The plant transformation is the not common in the efficient and transient development. These are the transgenic plants. Some of the transgenic plants What are the important of the these are the resistant to the fungal diseases, pathogens related proteins, chitinase, beta 1 3 glucanase, activation of the collagen, resistant to the bacterial disease, the antimicrobial peptides, lysozyme, ceropsin, sarcotoxin, engineering toxin, intelligence, restriction to insects, BT the toxin, Bacillus thuringiensis, BT plants, lectins, protease inhibitors, alpha amylase inhibitors, resistance to the viral diseases, resistance to the RNA approach, ribosome inactive proteins. These are the some of the resistant genes.